So our next speaker, the last of the session, is Nicolas Gustavo Calleja from the Institute of Biology and Experimental Medicine in Argentina. So we'll talk about the evolutionary roots of fear in amphibians. So Nicolas, you can now share your screen and start your presentation.
Okay. All of you can see it. All right. Well, thank you so much for accepting my presentation. And well, my name is Nicolas Calleja. I am a first year PhD student in the University of Buenos Aires. And now I'm going to tell you something about my grade thesis. In Argentina, we have like a final work for the first grade. And now I am making some improvements in this work for my PhD. A little route for this presentation is like a very short introduction, why we use amphibians and what is comparative psychology, what is fear for us, cardiac conditioning, that is my grade thesis, and the Ahinor technique, that is something that I am currently working. Well, for introduction, why use a comparative model to study fear? Well, I like this quote of Charles Darwin. I don't know if this one is real or not, but I really like it because it's a very good example of there is no fundamental difference between humans and animals in their ability to feel pleasure, pain, happiness, or misery. So we can say that any animal, human or not human, can feel emotions. Comparative psychology have the objective of comparing different species to identify common and divergent aspects of behavior, and in this way, we can understand the evolutionary story of different processes such as learning, memory, emotions, and all of a lot of things that we can see on behavior. As studying simpler brains, we can understand how it was the evolutionary origins of very highly complex processes. From Donjan of 2010, we can see 5 like biggest advantages of animal models. One of this is the greater variable control. We have a lot of control of variable because we create the animals, and we can study the biological basis. We can study simple brains, as I said before. The animals doesn't have the compliance with the investigator and doesn't have the linguistic processes along his brains. And I think the most important thing about studying fear in comparative psychology is the acquisition of negative behaviors. We don't know how a person can get a phobia, but we can make a phobia in animals. Well, why amphibians? The thing about the amphibians is that they are a very simple model of they have a simple model of brain. They have a simple nervous system which allowed us to study basic neural substrates, and in this way explore the evolutionary origins of fear response. We can think that the amphibian are the first terrestrial vertebrates, so it has a lot of changes in his brain that been from fish to the first mammals. It doesn't have a neocortex, so it doesn't have what we used to say modulation of behavior. So it's like a very simple model in vertebrates to study complex processes. Well, what is fear for us? Fear is a basic and evolutionary ancient emotion. It's an alarm system against danger, and is accompanied by physiological adaptation. Yes, it prepares us to face a threat. We can fight or flight with all this physiological energy, like increasing blood pressure, heart rate, sweating, increased muscle tone, and another changes that make us be ready for fight or flight. Yes, for being prepared for the for this stress or for this alarm. Here I'm going to tell you how is a condition and how is the conditional fear paradigm that has a conditional stimulus that in first place it doesn't make any response in the subject, but then we have an unconditional stimulus like could be the explode of a balloon. Yes, the balloon per se doesn't make any response in the baby, but when it explodes, it makes a big sound that makes the baby cry. So if I present the balloon and every time I present the balloon, I make it blow, I can see that after a few times, the baby, when he sees the balloon, he's going to cry. Yes, I can see that he seeing the conditional stimulus is going to make the fear response without the unconditional stimulus, without the explode. But also we know that we can extinguish this fear response by presenting the conditional stimulus repetitively without the unconditional stimulus, without the explode. If I present the conditional stimulus several times, I can make the extinction of this fear response. I can make that the baby doesn't cry when he sees the balloon. What is important about studying fear? Well, evolution against culture, I mean with this subtitle that there is a mismatch in the evolutionary story of our species and what the culture, the actual culture make us unleash these responses. This difference, this mismatch between evolution and culture make us a lot of psychological disorders like anxiety disorders, phobia, or a lot of cardiovascular diseases that make us a lot of have an unhealthy life. What is important about studying fear? Well, phobias are often treated with exposure therapies that are essentially extinction procedures, the thing that I explained before. Although the extinction produces behavioral and emotional effects, it doesn't reverse the effects of acquisition, and extinction does not erase what has been learned. Yes, a phobia is very difficult to erase totally, so there are a few phenomena like spontaneous evocation that make us have the same fear with only one presentation of the unconditional stimulus. Well, here I'm going to tell you something about my thesis about cardiac conditioning. What we made is take two different works, one from Cerviño and Rodriguez of 2017, and one from Daneri and Musio from 2007. In one we still or take the protocol to use a fear conditioning and a protocol that shows to work for amphibians, and in the other side we use electrodes to measure the heart rate of the amphibians. What we have to do in my thesis was like getting on point these two things by separate and mixed it and see what happened if we could record the heart rate of the amphibian along all this all the sessions of training in the bufo, in the Rinel Arenal. What we use as a stimulus? Well, a lot of work in my laboratory before I even get here are about behavior with different solutions, because we can use electroshocks to like an aversive stimulus in amphibians because the highly percent of water that his body has. So we use as a appetitive solution saline solutions. Yes, we use as a appetitive solution, we use a saline solution lower than 250 millimolar. We use as a neutral solution or a neutral stimulus at 300 millimolar solution, and we use as an aversive solution one solution that is bigger for 400 millimolar salinity. What does it mean? Well, a solution that has that is appetitive makes the amphibian gain weight. Yes, it makes the amphibian take water, and an aversive solution make it deshydrate. That is something very dangerous for the amphibians because almost his 80% of body are water. Well, the experimental apparatus, we use an escapable box with some membrane on the base of this box, so we can present the solutions with some bottles that have some poly systems, and we are going to present the different solutions in a sequence, making the amphibian perceive it from the behind. And the amphibian is going to be connected by electrodes in his back to an electrocardiograph that is going to record the whole training sessions. The heart rate variable is a very useful depend variable as an indicator of fear and anxiety, and there are a lot of examples from a lot of years that use this as an indicator of fear and anxiety. Well, here I'm going to tell you how was the acquisition procedure. We have three groups: the pair, the no pair, and the only CS. So what we see in the pair is a presentation of the neutral stimulus, then a draining, and then the presentation of the unconditional stimulus of 800 millimolar. So it's very saline. It's a very saline solution that makes the dehydration of the amphibian very strong. In the no pair group, what we do is like a delay in the presentation of both the stimulus. Yes, we make instead of 30 seconds of draining, we extend it to 180 seconds to delay between one stimulus and the other one, so we can make sure that the amphibian doesn't associate the conditional stimulus to the unconditional stimulus. And in the second control group, we present in both cases a neutral solution, a solution of 300 millimolar, so it doesn't have the unconditional stimulus, so we use it as a control that without the unconditional stimulus, the presentation of these solutions per se doesn't make the fear condition. Here are the results. Here we see on the left the acquisition training day 1. What we see is that paired and no paired group show an increase on hit rate when we present the unconditional stimulus, proving this that the solution of 800 millimolar makes an erase of the heart rate response. And in the right, we see the acquisition after 6 training days, 1 session per day. When what we note is that the pair group, the one that has to be the one that have to associate the conditional stimulus to the unconditional stimulus, makes an anticipatory tachycardia. Yes, we see that when he starts to perceive this neutral stimulus, it makes a very it has a very higher heart rate when the presentation of the conditional stimulus. Now I am going to tell you about the extinction procedure. The protocol stays almost the same, but the changes we are making is that in the pair group, we present an unconditional stimulus of 300 millimolar. Yes, we present again the neutral stimulus. So what we want with this is the extinction. Yes, if I took out the the unconditional stimulus, maybe it will make that this anticipatory tachycardia that we see in the training, in the 6th day of training, is going to be extinct. What we see on the left is the graph that we saw before, when we see the anticipation of tachycardia. And after 6 days of extinction, after 6 days without presenting the unconditional stimulus, we can see that all the lines of the heart rate are stay the same. So what does it mean? We made it. we get the extinction works. And also something very important about this study is that we have completed 12 days of training with the electrodes in the back of the amphibian. That is something very difficult, or was very difficult a few years ago. Now I am going to tell you something I have been made in my first year of the PhD. With the AGNOR technique, we make the AGNOR technique procedure. For the acquisition procedure, we only see difference between the first 6 days of training. So in addition to heart rate monitoring, we take some samples of the brains of the amphibians, and we staining to investigate neural activity. Here we see some images that how does the brain of the amphibian looks, and how we can see the nucleus and the nucleolus, so we can measure the activation of the different structures on the brain. What we see in the graph is with only preliminary data. We only have 6 animals, and from only 2 groups. We only compare the pair and the only CS groups because I have to make more analysis, but we see that we can find a difference between the amygdala and striatum, significant difference between these 2 structures that is very common in the literature. So it's like a very good first result for this work. And I am going to finish this, I think maybe for December. I have all the animals. I have 20 animals to measure and see what happens and see the neural activity, but here are some promising data. Well, thank you so much for the invitation. Thank you so much for being here and for listening. I'm sorry for my English. And this is my email and the Instagram of the from my laboratory, so all of you can be connected with us with all our themes.
Thank you, Nicolas. Your English was perfect. We don't have much time for question. I will just ask you one. If I don't see it's coming, but I don't see a question yet. So do you think you could see difference in fear responses between sex and age, like difference in the responses if it was male and female, or if it was old or young animals? Would you see difference in these responses?
Yeah, the thing about amphibians is that we don't know exactly the age of the amphibians, because we take it from the nature. And so about age, we see like a very different heart rate basis in each animal. So we use an index to measure to do the statistics. And between male and female is something that we are going to look about. We are going to try this maybe this summer or the next summer. Here in Argentina, we are in spring. Maybe this or the next summer is something that I would like to probe, see what happens, if it's different or if it's stay the same.
Perfect, thanks. I will ask you a few more questions since we have time, because you were the last speaker.
All right.
We have a question from Catherine. Hello, thanks for the talk. Do the frogs show a different behavior in reaction to the US, or it's just increase in heart rate?
I only make the study of the heart rate, but yes, it makes something behavioral. It tries to escape from the situation, obviously, and it's starting to jump and move around, but it doesn't have a lot of space. That's the unescapable box that we put in. We are going to the thing about the heart rate is that the movement makes some noise in the recording, so it's a little bit difficult to make some more behavioral studies with the heart rate, but it's something that I would like to explore.
Yeah, she was suggesting maybe an increase in respiratory rhythm.
Yes, it could be, yes.
And we have one last question from Hélène. Cool experiment. What is the fitness advantage of increasing neural activity in when experiencing fear? What would
Sorry?
what would be the fitness advantage of increasing neural activity when facing fear?
What will be something? Sorry,
What would
I
be the
didn't
fitness?
understand the first part.
Yeah, what would be the fitness advantage of increasing?
Okay. No, what we want to study in the second chapter of my PhD is like the what happens when we make different extinction protocols. So what we want to compare there is see if any other structure is used in another extinction procedures. So we use this like the baseline for any future studies, and then we are going to play a little bit with the protocols, with the cardiac conditioning and all of this. It's like a very preliminary work.
Perfect. So thank you again for this great presentation. I think you can join Discord. You will have more questions, and you can now leave the session. Thank you again, Nicolas.